Welcome to the Asian Longhorned Tick and Theileria orientalis in cattle webinar. We thank you for coming to this presentation. My name is Dr. Caitlin Macy, and I am the District 9 Field Veterinarian with the Board of Animal Health. And I'm also leading the state's Veterinary Medical Reserve Corps, which is involved in training, developing, and deploying animal emergency response teams. So today's speakers are Dr. Eric Renshaw from the Board of Animal Health and Lee Green from the Indiana Department of Health. And we also have a bonus Dr. Lamb here who can help assist us with questions. Dr. Renshaw is a 2000 graduate from Purdue University School of Veterinary Medicine. He joined BOAH in 2016 after spending the majority of his career in emergency and laboratory animal medicine. He is currently the District 10 Field Veterinarian, which is in southwestern Indiana, and also facilitates the One Health Zoonosis program at the Board of Animal Health. Dr. Renshaw works closely with the Indiana Department of Health. And Lee Green is the Senior Medical Entomologist at the Indiana Department of Health. Before taking that role in 2021, he was a field medical entomologist at the Indiana Department of Health for over 15 years, where he served as a vector-borne disease subject matter expert in northern Indiana. During that time, he collected and identified over 500,000 mosquitoes and 4,000 ticks to study the arthropods and diseases that impact human health. And again, Dr. Lamb is also here from the Board of Animal Health, a district veterinarian with the Board of Animal Health up in the northern portion of the state, and will be here to answer questions at the end. Thank you so much for joining today. And a couple of housekeeping items. Like I mentioned, the webinar is not able to type in your questions, but if you dial *6 at the end with some questions, if you have any, we would be more than happy to answer them. So thank you both for joining us, and take it away, Mr. Green.
Well, this will be fun. Trying to describe a tick to you guys without you being able to see the slides. But we'll do our best, and you can look at the recording later. First, to introduce you to the ticks of Indiana, we do have about 20 different species of ticks that exist here in the state. However, there are 5 of them that are commonly occurred by people, whether those be on us or our pets. The first one is the top left, which will be the American dog tick. This tick is very common in all 92 counties of the state. It has an affinity for dogs and is very common throughout the summer. So this is the one veterinarians will often pick off dogs in the summer months. On the far right is a brown dog tick, Rhipicephalus sanguineus. While we do not detect this tick in nature, it can become a huge problem indoors. This is one of the ticks that can survive indoors and therefore can become a problem in places like kennels or in clinics. This tick also, of our most commonly found ticks, would look to me to be the most similar looking to the Asian longhorn tick. Then have slides of the other 3 ticks. The first one is Ixodes scapularis. This is a tick with the Department of Health. We spend a lot of time doing surveillance for, because it is the one that transmits Borrelia burgdorferi, which would be the causative agent of Lyme disease. Through our active surveillance program, we have found this tick in every single county in the state now. Most of the time, people associate this tick with the northwestern part of Indiana or along the Wabash River. However, it is in every single county. And one thing to remember about these ticks are all of these ticks are 3-host hard ticks. And because they live in a 2-year life cycle, each life stage of that tick will feed at a specific time of the year. Therefore, very rarely will you find multiple life stages on an animal or yourself. As with scapularis and most of the ticks we find, the larvae will feed on small mammals in the summertime. Once they feed, they'll drop off and molt into nymphs. and then become dormant all the way until the next spring. Here in Indiana, the nymphs will actively look for a blood meal in quest right around Memorial Day, which is not good news for public health. That's one of the most popular weekends for outdoor activities, and that's when that nymph is most actively looking for blood meals. They'll get on both small mammals and large mammals, feed, drop off molten tuna adults, and then the adults are most active when deer are active. So right now, those adults that will be searching for blood meals in the fall, when they do find a blood meal, they often will mate on host, drop off. The males will then soon die. The females will then lay eggs and die. If that tick does not find a blood meal this fall, it will become active and search for blood meals anytime it's above freezing through the rest of the winter and into spring. So that's why you won't see multiple life stages on a single animal most of the time. The next tick would be Amblyomma americanum. This is the Lone Star tick, very common in the southern third of the state, with another population along the Great Lakes region. This tick is a very aggressive biter. Oftentimes, especially ungulates in southern Indiana, you will find a large tick load of these on them. The females are very easily identified by a white dot in the center of their body. That's why they are called the Lone Star tick. And they have very long mouth parts. The third tick, or the fifth tick that we are just now finding in the state, is Amblyomma maculatum, which is the Gulf Coast tick. As the name suggests, this tick is most commonly found along the Gulf Coast, but it has expanded its region in the last few years, and we are now discovering it in pretty large establishments in southern Indiana. So that brings us to a brand new tick that we are looking for, which would be Haemaphysalis longicornis, or the Asian longhorn tick. As a little background, this tick is originally indigenous to Northeast Asia. It's an Asian tick. And in the early 1900s, this tick expanded into Australia and New Zealand due to the cattle trade between Australia, New Zealand, and Japan in the early 1900s. There, this tick has become a big problem in the cattle industry. It is a very aggressive biter, and especially in the moister environments of Australia and New Zealand along the coast, it has become a problem to their cattle industry. It is, again, a three-host hard tick, just like Blackleggeds and Lone Stars here in Indiana. But this tick has two different types of clades. There can be two different populations: a bisexual population, which has a normal 2-year life cycle, just like I explained in Ixodes scapularis. However, that's not the tick we have found in the U.S. We are finding the parthenogenetic version of this tick, which means the female does not have to mate in order to reproduce. She will simply make copies of herself, and therefore does not need to mate in order to reproduce. And because of that parthenogenetic population, they breed much faster, and that's one way we can distinguish this tick from the others, is oftentimes on animals you will find multiple life stages. So let's talk about Asian longhorn ticks in the United States. Our story begins in 2017 in New Jersey. What happened was a woman went to her vector control agency complaining about ticks. She said there was ticks all over her and her animals. Luckily, the person at that vector control agency was pretty familiar with ticks, knew this didn't make sense, and wanted to look at it himself. So they visited her property and found that her sheep were absolutely loaded with multiple life stages of ticks, took some samples, and discovered that they were Haemaphysalis longicornis. That was the first detection outside of a quarantine facility in the United States. However, now that we've become familiar with this particular tick, NVSL went back through some of their archives and had some larvae all the way back in 2010 in West Virginia. That's part of this tick is Haemaphysalis longicornis. Actually has some cousins that we do have here in the United States. One of those would be Haemaphysalis leporus palustris, which is commonly called the rabbit tick. So it is kind of hard to identify these ticks. Now, we have found Asian longhorn ticks in 17 different states, mostly in the eastern U.S. As you can see from this map, down the eastern seaboard, they're finding these ticks everywhere. And unfortunately, they are now in 2 of our neighboring states. Ohio has found longhorn ticks in 7 of their counties, and Kentucky has found longhorn ticks in 8 of their counties, including Boone County, which is just across the Ohio River from Dearborn County, Indiana. Through the increased surveillance across the United States, this is the different animals that this tick has been discovered on. As you can see, environmental, which means people physically going out and looking for ticks and conducting tick drags, has been the most prevalent. But domestic animals are often found with this tick on them. The cool thing about this graph is you can see this tick follows kind of the same pattern as far as going into diapause during the winter as our common ticks do. So there is still going to be a tick season with this from about March to October. Here's just a different way to show this data. As you can see, there's all kinds of different wildlife that this tick can get on. They really do like ungulates, so they're most commonly found on white-tailed deer. But as you can see here, they're found on mammals that won't help with their travel very much, such as Peromyscus or white-footed mice. But some other animals that do have pretty broad ranges, such as white-tailed deer, to help spread this tick across the United States. It's also been found on several different species of birds, some migratory and some non-migratory, which will definitely help the spread and why we are so interested in the surveillance. So it's been found on hawks, owls, geese, as well as some other birds. As far as people and animals goes, it does prefer to like ungulates, but it has now been found on every commodity animal, cats, dogs, sheep, horses, goats, chickens, pigs. It does not seem to like humans, although it is found on a bunch just because we are so cognitive of ticks on ourselves. So this is why we are imploring vets to help with our surveillance. Another part of this tick is because it's parthenogenetic and doesn't need a male to lay eggs, populations of this tick can explode in areas. Tick populations can be so large and the tick load so big on an animal that it could exsanguinate and kill livestock. We have seen that happen in situations with cows in Virginia, Tennessee, North Carolina, and as close to home as Kentucky and Ohio. Healthy animals can be exsanguinated by this. So that's part of the surveillance process would be if you do see an animal with an extremely high tick load, that would be a tick that we are very interested in looking at. As far as this tick transmitting disease, the good news is it doesn't seem to transmit human disease. In the lab, it's not a competent vector for Lyme, human granulosis, anaplasmosis, tularemia. In the lab, it is a competent vector of Rocky Mountain spotted fever, heartland, and Powassan virus. And most importantly, of your interest was the U.S. origin parthenogenetic Asian longhorn tick will vector Theileria orientalis in the lab. As far as field collected disease pathogens within those ticks, we have found cattle Theileriosis from Virginia. There has been a tick tested in Pennsylvania that had Lyme disease. Also anaplasmosis and bourbon virus from Virginia. It's important to keep in mind that ticks feed on blood. So oftentimes ticks are really just bags of pathogens. So just because a pathogen is discovered within them does not mean they can turn around and transmit it. An important distinction to keep in mind. Finally, here is some modeling that has been done, and thank you to Denise Bonilla from the USDA who has shared these slides. This is some modeling that has been done of where we potentially think longhorn ticks could establish, and Indiana is one of those states. It could be a problem. So how do we ID? Some key characteristics of this tick if you want to identify it. The most important characteristic is the palps of this tick do extend laterally. They look very triangular if you look at the tick. There is another tick, that Rhipicephalus sanguineus. If you can look on the far right, right here, oftentimes this hexagonal shaped basis capitulum can be mistaken to be triangular and extending palps. That's not true. What we are looking for here on the left are those extension of the palps that make it hemophysalis. It does take some pretty good microscopy to separate Hemophysalis longicornis from other Hemophysalis, such as Leporus palustris. So if you do have any tick that looks like this, we would be very interested to look at it and see what we can find. So this is the tick itself. You can see these extended palps, pretty easy to see with the naked eye, and should alert you to call us as part of our surveillance system. If you do see that, keep in mind that there are several similar looking ticks. On the left here is a longicornis nymph, where you can see its palps, versus down in southern Indiana, the very common Lone Star tick nymph, which is a very aggressive feeder you will often find on animals. You see those long skinny palps as a dead giveaway the difference between the two. So if you do have a tick you think would qualify as a hemophysalis, we are very interested in that. To submit ticks, preserve those in 70 to 95% ethanol, and then contact your BOAH district veterinarian for identification. If that does not work or not an option for you, you could always mail them to our lab. Obviously, we would want the information, what it was collected on, and the location. That way, if it is a longicornis, we could do some follow-up visits. And I think that is it. And if you have any questions, we can save those for the end.
Okay, good afternoon. As Dr. Macy mentioned, my name is Eric Renshaw. I'm the District 10 field veterinarian. I also have the One Health and zoonotic disease programs. And as a part of that, I participate on the tick-borne advisory committee, and that's where I met Lee Green. This committee has been talking about the longhorn tick for several years, and I think it's very relevant for veterinarians across the state to learn more about it. I will be talking about Theileria orientalis specifically. So what is Theileria orientalis? It's a tick-borne obligate intracellular protozoan. It infects both red and white blood cells of cattle. The estimated cost to the cattle industry is around $300 million a year, and $400 to $760 per animal, and that accounts for abortions as well. It is the cause of bovine infectious anemia. Fortunately, T. orientalis does not affect humans, and the meat from T. orientalis infected cattle is not a human health risk. It's only known to be pathogenic to domestic and wild cattle. Once thought to be a foreign agent, but now, unfortunately, it's spreading quickly across the U.S. So what are the regulations surrounding Theileria? Currently, there are no movement restrictions for positive cattle, nor those cattle that have been exposed to the Asian longhorn tick. It is not reportable in Indiana or to the USDA, whereas Theileria annulata, Theileria parva are reportable. These are considered transforming diseases that lead to lymphoma, and that's the difference. For Theileria orientalis, there are 3 different genotypes, and this is pretty important. The first one is Ixodes. So Ixodes has been discovered in Virginia, West Virginia, Tennessee, Kansas, Pennsylvania, Maryland, North Carolina, Kentucky, and New York. And this one is the virulent or the pathogenic genotype. So Ixodes first showed up in the U.S. in August of 2017 in a Virginia cattle herd. They had some unexplained deaths. After doing some diagnostics, they determined it to be Theileria orientalis Ixodes. Since it was discovered in Virginia, Maryland, and Virginia Tech colleges of veterinary medicine, they basically led the way in research publications and are still doing so. The second genotype is Ixodes. It's been diagnosed in Virginia, Texas, Louisiana, Vermont, Wisconsin, and now Indiana. And that's kind of the genesis of this talk today. And this is considered to have variable pathogenicity. The third and final is Buffalo. And so it's been found in Texas and the states that surround Texas since 2000, and is considered the benign genotype 1, not to worry about as much. So far in the U.S., multiple genotypes have not been diagnosed in the same animal, whereas in other countries and continents, it has been. So transmission, it can be by mechanical transmission since it's a blood-borne pathogen. Needles, lice, biting flies, transplacentally, which is rare, and most importantly, the saliva of the Asian longhorn tick. If it's an Asian longhorn tick that transmits it, it's going to be more rapid, and the disease progression is going to be quicker than if it's by mechanical means. Unfortunately, once an animal is infected, it's going to be persistently infected for life, and it will serve as a reservoir for Theileria orientalis. After the animal gets through the acute phase, however, the number of organisms decreases dramatically, meaning 1,000-fold even. So another important topic is the life cycle. So cattle are typically infected within 3 weeks of being placed on a pasture with tick vectors. The Theileria sporozoites, which are the infective stage, are transmitted through the tick's saliva. And as quickly as within 10 days, that same cow can then be infective to other ticks. The Theileria mature within the tick after attaching to the host within 2 to 3 days, et cetera, et cetera. As you can tell, it can move pretty quickly. Grossly infected cattle may have hundreds to thousands of the Asian longhorn ticks, as Lee mentioned, and they can be in multiple life stages. And unfortunately, the Asian longhorn tick population can explode. So that's the concern here. The ticks can remain in a pasture for up to 2 years. And most of the diagnoses that have been made in the eastern part of the country have been made in spring and fall. Now, is this because there's more submission with the calving, or is it the activity of the ticks? And that's still to be up for discussion. So symptoms, for those of you that have seen anaplasmosis, the symptoms are going to be very, very similar. The acute phase of the disease is 1 to 8 weeks after becoming infected. A large percentage of these cattle will have no clinical signs at all. The big clinical symptom is going to be anemia. They can be lethargic, pyrexic. They're going to be yellow. Some people call them banana cows. They're so yellow. Ventral edema, late-term abortions, and calf loss. So this is the one feature that's going to be dissimilar to anaplasmosis, that it can actually kill calves. As mentioned, many herds are asymptomatic, but the death loss rate is 1 to 5% in the literature, but it can be variable, and it's been reported as high as 30%. Another key point to remember is that death is often linked to stress. So as I mentioned with calving, if they're not getting fed enough, if they don't have access to water and handling. Majority, thankfully, have limited or no clinical signs. Not all clinical disease in a positive animal is a result of theileriosis, and many times they're going to be co-infected with anaplasmosis. The point to drive home here is if they're anemic, think Theileria. Once they get through the acute phase and they survive that, there is no obvious long-term health or production effects of the persistent infection. So that's one positive. And then once they get through it, they are protected from the subsequent clinical disease, but they will forever be a carrier. So testing, PCR is the gold standard, and that's going to be how you determine what genotype, whether it's Ikeda, Ketosi, or Buffalo. So for submission, it's going to be whole EDTA blood, spleen, liver, kidney, or lymph node samples. For those of you that are proficient at performing blood smears, you can also diagnose it with blood smears and the appropriate stains. Treatment. Unfortunately, there is no approved treatment, and at this time, there's no vaccine in the U.S. Buparvaquone has been shown to be effective in treating cattle in the early stages, but not for those that are already anemic. Theileria has been endemic in Australia for 20 years. This drug is still not approved here, and it's unlikely to be approved in the U.S. So again, no specific treatment. Really, it comes down to supportive care, blood transfusions, B vitamins, fluid therapy. But the main thing, if you have the diagnosis or you're suspicious of it, is to minimize stress. I think this is all related to a decreased oxygen carrying capacity. If they're anemic, they can't handle any stress at all. So that's another key point. So how do we prevent this? Not easy. It comes down to, any other disease that's related to ticks. So it's tick control, pesticide treatment, whether it's pour-on or pesticide impregnated ear tags, visually inspecting your cattle for ticks and removing them, which is not always practical, keeping your pastures mowed short, keeping cattle out of wooded areas, and restrict movement of grazing cattle. So if you know you don't have it in a specific pasture, you may want to keep them there if this disease is in your area. There are no available acaricides in the U.S. that carry a label specific to the Asian longhorn tick. In some literature you read, there's variable efficacy anyway, because a lot of times the Asian longhorn tick is going to be in the environment and not on their biologic host. For this audience, if and when this becomes a bigger problem, single use of needles is going to be important to remember because it is transmitted by the blood. Cleaning and disinfecting of tattoo and dehorning equipment between animals. And then if you were to purchase any animals in an endemic area, you'd want to have those cattle tested for Theileria and then visually inspect those cattle for the Asian longhorn tick as well. So this entire webinar is somewhat predicated on a Southern Indiana case study. A 6-year-old crossbred cow presented dead to SciPac, which is the Southern Indiana Purdue Ag Center, and this was in early September 2022. The one thing the producer noted antemortem was that the cow was recumbent, so very weak. Grossly, it was emaciated, it was yellow, icteric, and it had an enlarged spleen. So the pathologist was astute enough to call for a Theileria and an Anaplasma marginale PCR test. It was positive for both, so they needed to determine what genotype of Theileria. They submitted a splenic sample, and it was positive for Theileria chetosi, but negative for Ixodes. So keep in mind, if you remember from the beginning of the talk, Ixodes is the pathogenic form, whereas the chetosi is kind of more mild as far as pathogenicity. So likely this cow in Southern Indiana that was positive for Theileria likely died from anaplasmosis. Don't know for sure. But what is noteworthy is we are now the 6th state to have the chetosi genotype, diagnosed. So that begs the question, is the Asian longhorn tick in Indiana? We've just not found it yet. And to summarize, for veterinarians, since this somewhat originated in Virginia, the Virginia Cooperative Extension has a lot of good information for your cattle producers. For those of you that enjoy YouTube videos, the National Cattlemen's Beef Association had a 2-day webinar on ticks on day 1 hour, 57 minutes in on YouTube. There's a great presentation that was given by Dr. Kevin Lammers, and he's kind of the leading authority in the U.S. We're attempting to collaborate with Dr. Lammers in Indiana. He's interested in this herd in Southern Indiana and performing blood draws this spring to determine the prevalence of the Theileria chetosi. So I'm going to hand it back over to Lee, because once we had this diagnosis, we were interested, the Department of Health was interested, and a tick drag was conducted by Doug Ginder, who is the Southern Indiana entomologist, and Lee's going to give us an update on where that stands.
So when we found out about this case, Doug went to the farm and actually conducted tick drags through the entire pasture. When he did that, it was getting into mid-October, so we were worried that the Asian longhorn ticks had already gone into diapause. However, with talking to Ohio State University, they were collecting ticks much later than Doug did the drag there in Southern Indiana. So at least in Ohio, the ticks that we were looking for was still active. When he did the drag, he found several different species. He found several Amblyomma americanum Lone Star ticks. He did find one black-legged tick right along. There's a woods that borders the pasture in that Ixodes area, and heart dropped when he did find one single Hemophysalis Leporus palustris. So the whole drive home, he was worried that he had found the first longhorn tick, but it turned out to not. We do plan to drag again this spring to just double check and see if we can find any Asian longhorn ticks, both in this particular pasture, but since SciPac's pretty close, drag in some other areas as well. Those ticks were submitted for PCR testing for Theileria orientalis Katosi, just to see there's so little known about that disease. If any of those ticks vectored it. I haven't heard any results yet, so either they tested negative or they are still in queue to be tested. So that's where we're at. Indication we have that the Longhorned tick is in the state. We will follow up on because this is a big deal.
Thank you so much. Thank you both so much for this awesome presentation.